In this video, I'm going to go through some terminology that's relevant to conjugation in bacteria. And this has to do with different types of cells and names that we call them, okay? So let's say we're looking at one cell here, okay? This is the bacterial cell. Inside this cell, we have the bacterial chromosome. And just like we went over in the first video of this lecture series, bacteria have just a single chromosome that is circular, okay? Now, in addition to this one circular chromosome, bacteria often will have what we call extra chromosomal DNA. So this is little molecules of DNA, typically pretty small, that are separate from the genome and not considered part of the genome, okay? The type of extra chromosomal DNA that we're going to talk about today, and I'm going to color this in red, this is called a plasmid, okay? So where a chromosome might be like a couple million base pairs long, a chromosome might just be a couple thousand base pairs long, okay? So orders of magnitude smaller. And let me shift this over a little bit here, okay? The specific type of plasmid that I'm going to talk about here is a plasmid that we call the F plasmid, or the F factor, okay? And we call this F because it confers fertility on the bacteria. It allows this bacteria to initiate gene exchange with other cells, okay? And bacteria that have this F factor as a separate plasmid, we call these F+, okay? Now, I'm going to erase, well, let me say that again. An F+ cell would probably have many copies of that F factor plasmid, but I'm going to erase some of these here, and let's just focus on like one copy of a plasmid just to give us some space to write, okay? So putting in our one plasmid here, let's put that here. This is the F plasmid, okay? And now, not all cells have the F factor, have that F plasmid. You could have another cell that would have the bacterial chromosome, okay, and it doesn't have any F factor plasmid. So no F plasmid, and we call that cell F-, okay? Now, this F+ cell can initiate a process called conjugation with the F- cell, okay? And when it does that, it actually forms a physical connection between the two cells, a little canal, a little tube would be a better way to put that. And it's going to physically connect one cell to the other. This is a structure called a pilus. Okay? And when that happens, our F plasmid is DNA, it's double-stranded, so I'm going to draw both strands on here, okay? In one of those strands, we're going to get a little break. So a little break in the sugar phosphate backbone there, okay? And then this one strand that has a little nick in it is going to travel through the pilus into the F- cell, into this recipient cell, okay? And we can get the entire F factor transferred over from the F-, or at least one strand of it, from the F+ cell to the F- cell, okay? And when that happens, then the F- cell, or the cell that was F-, will make a strand that is complementary to the strand that transferred in, okay? The strand that was lost up here in this original F+ cell gets replaced. And so now we're going to break this connection here. The pilus doesn't last very long, okay? So we're just going to break that connection, lose our pilus, okay? And we end up with our F plus, I'm sorry, that should be in red. Our F plus cell remains F plus, and our cell that was F minus becomes now F plus. Okay? So that's one way the bacterial, what we call bacterial mating or bacterial conjugation can happen. Okay? Now, that's not the only way. Now, you'd think that might be the only way, right? Because you could either have an F factor or you could not have an F factor. But that's actually not the only two conditions. Okay? So let's go back to our condition here of having an F minus cell. Okay, and I'm going to scoot this stuff up a little bit. Okay, so let's scoot this up. I'm going to draw in a new cell here. Okay, and it has a chromosome. Actually, let's make that chromosome a little bit bigger than that because this is the important part of this cell here. Okay, we're going to have a chromosome here, and on that chromosome, we are going to have the F factor incorporated into the chromosome. Okay, so this is our chromosome with F. Okay, so this red part of the chromosome here on this third cell is the F factor DNA incorporated into the bacterial chromosome. And so we call this type of cell HFR. Okay, capital H, little f, little r. And that stands for high frequency of recombination. There. Okay, now, this HFR cell having the F factor admittedly incorporated into the chromosome, but it still is there. It can also initiate bacterial mating. It can initiate conjugation. Okay, and so it does it the same way, right? It forms this pilus. Let's have our F minus cell receive DNA again. Okay, and make a little opening in our cells here to allow the DNA to get through. Okay, now what's going to happen here is our bacterial chromosome is also double-stranded DNA, right? Let's make two strands there. And our other strand where the F factor is in red. Now, what's going to happen is we're going to, again, make a little break in our double-stranded DNA, okay, right on one side of the bacterial chromosome, or on one side of where the F factor is in the chromosome, okay? And actually what's going to happen is, right on one side of this break, we have the F factor DNA. On the other side of the break, we have the bacterial chromosomal DNA. It's actually the chromosomal side of that break that starts to move through the pilus, okay? So this moves through here, and if we had some genes right next to that F factor, let's say we have genes A and B and C, and we have wild type copies of genes A, B, and C on this HFR chromosome, we could get some of that bacterial chromosome moving in here, right, with our genes A plus, B plus, and C plus, okay? Now maybe on this F minus chromosome, we would have the same genes, right? But we might have different alleles. So we might have A minus, B minus, and C minus, mutant versions of those three genes, okay? And what can happen is eventually these cells get like physically jostled. And so this pilus connection between cells doesn't last for super long, okay? So eventually it'll get broken, and we will lose that connection between the cells, okay? And so what's going to happen is, let's make a little Let's neaten this up a little bit here. Okay. We're going to move some of our bacterial chromosome DNA from our HFR cell, our donor, to our F- recipient. The last part of the DNA, because the DNA would be donated in, as I've drawn it, an anti-clockwise fashion or a counterclockwise fashion. We would get first the A+ gene, and then the B+ gene, and then the C+ gene move in, and we might get more genes than that depending on how long that connection lasts. But the last thing that would have come in, if we could have maintained the connection long enough, would be the F factor itself. Okay? But we didn't maintain that connection long enough. In fact, all we were able to transfer over was 3 genes. Okay? So we have these wild-type versions of these genes, A+, B+, and C+, came from the HFR cell, a strand of it, into the F- cell. And then what can happen is this homologous region from the chromosome to this new piece of DNA, we could get crossing over here, right? And get these wild-type genes now incorporated into the F- chromosome. Okay? So we can exchange genes in that way. Now, another thing that we often see is in an HFR cell, the F factor is incorporated into the chromosome, but it can also recombine out of the chromosome, okay? So if we get, like, the chromosome just kind of folds over on itself, right? And we get that F factor DNA just kind of excluded out of the chromosome, okay? Then what we have is we could potentially create a cell that would have, here's its chromosome. It doesn't have the F factor in the chromosome anymore. The F factor is instead now outside of the chromosome, but sometimes this process here can happen inexactly. So we don't precisely get only the F factor cross out of the chromosome. Instead, we can get a little bit of chromosomal DNA incorporated in here too, and we might have like the A+ gene in here now in the plasmid rather than in the chromosome. Okay, this now is called an F' cell because it has the F factor. It is outside, the F factor is outside of the chromosome, and it also has a little bit of the bacterial chromosome included in that plasmid. Okay? So these are some different terms that you'll need to know when we talk about conjugation.